Welcome to Integrative Bioanalytics. Today, we will guide you through our single-cell RNA-seq analysis workflow. To start your analysis, simply fill out our analysis form. Please indicate the chemistry used and whether your samples are cells or nuclei. You will receive a link to upload your data and submit metadata. Use the link in your email to notify us when your upload is done, and we will begin processing your data. First, we perform quality control. We reduce background contamination and batch effects and exclude low-quality cells and doublets. You will receive a comprehensive QC report, complete quality metrics, and visualizations. If the experiment QC fails, you may cancel the analysis at no cost. Now let's look at the analysis you will receive. First, cell clustering. Clustering is done at 3 different resolutions so you can choose the best level of detail for your research. Check the cluster tree to understand how clusters are further divided at higher resolutions and examine the bar plots for population compositions. Each cluster has associated marker genes. We include multiple visualizations, heat maps, dot plots, violin plots, and feature plots. To explore your data easily, use the interactive browser. You can check the expression of all your genes and even create your own clusters. All this data is integrated in a cluster report where we include AI-assisted cell type identification. We will work with you to refine the final clusters for downstream analysis. Check this video for more information about the process. The analysis also includes a curated Seurat object for RStudio and instructions to use it. Once clusters have been established, we perform differential analysis. Heat maps and interactive volcano plots are included. We compare everything you may need, one cluster against the rest, conditions against each other, and a cluster in one condition against the same cluster in another condition. All these permutations are also used for downstream analysis. Pathway analysis is performed using 3 different methods. We first quantify the baseline pathway activity of each group to understand the core underlying processes. Then, to characterize how pathways change between groups, we look at signatures and ontologies enriched in the comparisons. Transcription factor analysis follows the same approach. For each group, we quantify the baseline transcriptional activity of more than 1,000 transcription factors and regulators. Then, we analyze changes between groups for gene regulatory networks and enriched motifs at the promoters and enhancers of differentially expressed genes. We infer cell cycle by looking at gene expression. The results are shown with bar plots and the UMAP. Then, we look at cell signaling. The expression levels of ligands and receptors are used to calculate communication flow and identify specific pathways and signaling proteins. For RNA velocity, we obtain useful parameters such as latent time, rate of change, or the typical streamlines. Finally, you will receive a biological report with analysis highlights and detailed methods to help you with manuscript preparation. Every analysis includes follow-up time for revisions and minor changes. Visit Integrative Bioanalytics and experience bioinformatics made simple.